Rahim. My name is Kashaf Hassan and my department is BSBC, BS Biochemistry. And now I represent my presentation topic, and these are the contents of my presentation topic. Introduction, history, structure, function, types, and DNA sequencing. Then principles, procedure of DNA sequencing, advantages and disadvantages. Methods of DNA sequencing, RNA structure, function, types, discovery, DNA components and backbone, RNA structure, functions, types, and discovery, difference between DNA and RNA, DNA modification, types of DNA modification, what happens when DNA is damaged, summary and preference. And now, first of all, we see that the introduction of DNA sequencing. Basically, DNA sequencing is a laboratory method that is used to determine the sequence of DNA molecule. And the DNA was developed by Frederick Sanger in 1975. He was later awarded the Nobel Prize in Chemistry in 1980s for his contribution to understanding DNA sequencing. The story of DNA begins when Watson and Crick discovered the structure of DNA in the year 1953. In 1964, Richard Hawley, who performed the sequencing of the transfer RNA, was the first attempt to sequencing the nucleic acid. The structure of DNA sequencing, basically DNA is a double helical structure that is antiparallel and a complementary strand having a phosphate sugar backbone with nitrogenous bases stacked inside. Now we discuss the function of DNA sequencing. DNA sequencing is a laboratory technique used to determine the exact sequence of bases, which includes adenine, cytosine, guanine, and thymine in a DNA molecule. The DNA basis sequence carries the information a cell needs to assemble protein and RNA molecule. DNA sequence information is important to scientists investigating the functions of genes. Basically, DNA sequence comprises of two methods, in which first is Maxam-Gilbert and the second one is Sanger sequencing. Now we discuss the principles of DNA sequencing. Chain termination sequencing involves the synthesis of new strands of DNA complementary to a single-stranded template, and the process of determining the order of the nucleotide bases along a DNA strand is called sequencing. Now its procedure of DNA sequencing. Firstly, DNA amplification. Now it is PCR purification and then sequencing preparation. And now we discuss some procedures in which included PCR amplification of target sequence and amplicons purification, sequencing preparation, DNA sequencing and data analysis. Now it's the advantages of DNA. No premature termination due to DNA sequencing, so no problem with polymerase to synthesize DNA. And then the stretches of DNA can be sequenced which cannot be done with enzymatic method. And while the other side, the disadvantages of DNA sequencing. Now, not widely used, use of radioactivity and toxic chemicals. Basically, DNA sequencing method is historically. There are two main methods, as we said earlier, in which included firstly Maxam-Gilbert method and the second one is Sanger method, means Sanger sequencing. Modern sequencing equipment uses the principles of the Sanger techniques. Now we discuss the DNA. DNA is a chemical name for the molecule that carries genetic instructions in all living things. The DNA molecule consists of two strands that wind around one another to form a shape known as a double helix. Each strand is a backbone made of alternating sugar and phosphate groups. What type of chemistry in DNA? Basically, DNA means deoxyribonucleic acid is an organic chemical in which contains genetic information and instructions for protein synthesis. It is found in most cells of every organisms. Now it's the types of DNA. Basically, there are two types of DNA in the cell. in which include autosomal DNA and mitochondrial DNA. Autosomal DNA, also called nuclear DNA, is packaged into 22 paired chromosomes. In each pair of autosomes, one was inherited from the mother and one was inherited from the father. Now it's the structure of DNA. As you say earlier, DNA sequencing was discovered by the James Watson and Francis Crick. And now it's also included in the first discovery in 1869. But its role in genetic inheritance was not demonstrated until 1943. In 1953, James Watson and Francis Crick, aided by the work of biophysicist Rosalind Franklin, was determined that the structure of DNA is a double helix and the polymer, a spiral consisting of two DNA strands wound around each other. The breakthrough led to the significant advances in scientists' understanding of DNA replication and hereditary control of cellular activities. Basically, DNA is the genetic material and it's control the metabolic activities of the cell through which the protein synthesis. An error during DNA replication results in a change of genetic information or mutation. DNA along with protein histone from the chromosomes. Now it's the discovery of DNA. DNA basically was discovered in 1869 by Swiss researcher Frederick, who was originally trying to study the composition of lymphoid cells, means white blood cells. Instead, he isolated a new molecule he called nuclein from a cell called nucleus. So basically this is the structure of DNA. It included phosphate group, nitrogenous bases and deoxyribose sugar. And this is also a structure of DNA sequencing, and we say called backbone of DNA. It included deoxyribose sugar, phosphate groups and nitrogenous bases. And now we discuss the topic of RNA. RNA basically ribonucleic acid. RNA is a linear molecule composed of 4 types. It included adenine, cytosine, guanine and uracil. Now it's the types of DNA. Basically there are the 3 types of RNA exist. It included messenger, ribosomal and transfer. Now it's the structure of RNA. Basically RNA consists of 4 nitrogenous bases. It included adenine, cytosine, uracil and guanine. Uracil is a pyrimidine that is structurally similar to the thymine, another that is found in the DNA. Like thymine, uracil can base pair with adenine. Now this is the table of RNA. Different kinds of RNA included transfer RNA, ribosomal RNA, messenger RNA, small nuclear RNA, small interfering RNA and microRNA. Basically transfer RNA have small size and that transport amino acid to site of protein synthesis. And the ribosomal RNA is a several kinds variable in size. That's the combined with proteins to form ribosomes, the site of protein synthesis. And the messenger RNA, basically it's a variable in his size. That is direct amino acid sequence of proteins. Small nuclear RNA, basically small in size, that is process initial messenger RNA to its mature forming eukaryotes. And small interfering RNA is also small in size, that affects gene expression used by scientists to knock out a gene being studied. And microRNA is also small in size, that affects gene expressions. Basically, the primary functions of RNA is to create a protein via translation. RNA carries genetic information that is translated by ribosomes into various proteins necessary for cellular processes, in which messenger RNA, ribosomal RNA. and transfer RNA are the three main types of RNA involved in protein synthesis. RNA was also discovered means that the discovery of RNA begins with the discovery of nucleic acids by Frederick in 1868, who called the material nucleon since it was found in the nucleus. Now it's the table of DNA and RNA. You can see that DNA have some few points and also RNA have few points. Has the sugar, DNA has the sugar, the oxyribose, and RNA has the sugar ribose. DNA has the base of thymine and RNA include uracil. DNA is a double-stranded and the RNA is a single-stranded. DNA basically is a double helix and RNA does not form a double helix. The nitrogenous bases consist of DNA included adenine, guanine, thymine and cytosine. And the RNA included the nitrogenous bases consist of adenine, guanine, uracil and cytosine. Now these are the structure of DNA and RNA. So now you can say that these are the difference between the DNA and RNA as we said earlier. Have some differences, DNA and RNA. Now it's the DNA modification. DNA modification is epigenetic changes and DNA methylation. The addition of a methyl group or a chemical cap to part of the DNA molecule which prevents certain genes from being expressed. And histone modification is another example. Histones basically are also types of proteins. That's the DNA wrap around. Types of DNA modification. Basically, there are two types of epigenetic modification included DNA methylation and histone modification. Histone is also a type of protein synthesis. Now these are some structures which include cytosine, 5-methyl cytosine, 5-hydroxyl methyl, 5-formal cytosine, carboxyl cytosine and others. Now here is the questions arise. What happens when DNA is damaged? So DNA damage is a change is the basic structure of DNA that is not itself replicated. Means that DNA doesn't dependent. DNA is not itself replicated. When the DNA is replicated, a DNA damage can be a chemical addition or disruption to a base of DNA, creating an abnormal nucleotide or nucleotide fragment or a break in one or both chains of the DNA strand. Now it's a summary of DNA sequencing. Basically, DNA sequencing is used to determine the sequence of a DNA molecule. RNA contains the sugar ribose while the DNA contains the slightly different sugar deoxyribose, a type of ribose that lack one oxygen atom. And then B is RNA has the nucleobase uracil while DNA contains thymine. These epigenetic processes often dynamic chemical modification to DNA or to the histones, DNA modification. And now these are the preferences of my contents of my topic. So now thank you. Okay, it's all of my topic, and now I've wanted my topic. Okay, thank you.